Professor Anthony Hudetz is Professor of Anesthesiology, Physiology, and Biophysics at Medical College of Wisconsin, Milwaukee, Wisconsin. Dr. Hudetz graduated from the Eötvös Loránd University, Budapest, Hungary in 1974 with a MS degree in physics. He received a Doctor of Doctor of Medicine degree from the Semmelweis University of Medicine, Budapest, Hungary in 1979, and PhD from the Hungarian Academy of Qualifications in 1985. Dr. Hudetz has held faculty appointments in the Semmelweis University of Medicine and Louisiana University also. He is adjunct professor in the Department of Biomedical Engineering, Marquette University. At Medical College of Wisconsin, Professor Hudetz rose to the rank of professor in 1998 and received tenure in the Department of Anesthesiology in 2007. He is the recipient of the first Melvin H. Knisely Award from the International Society of Oxygen Transport to Tissue and a research award from the Hungarian Physiological Society. He is past president of International Society of Oxygen Transport to Tissue. He has published over 100 articles and has maintained NIH funding since 1997. His current research interest is in discovering the mechanisms by which anesthetics produce loss of consciousness. Does consciousness really disappear when the patient is not responding? What happens to the thinking brain when it is anesthetized? Please welcome.
Thank you very much for the kind introduction, and I greatly appreciate the invitation from Dr. Visal and the organizers, and Dr. Stuart Hameroff. It's wonderful to be here. As my title shows, I'm going to talk about anesthetic mechanisms, but I'm focusing also on consciousness as a means to better understand the relationship between anesthesia and neural correlates of consciousness in terms of dynamic brain states. Obviously, anesthesia has been a great mystery for 150 years. We still don't know how anesthetics work. We would love to know how anesthetics work because it would help the anesthesia profession further, but also it would highlight and it would enlighten us about the mechanism of consciousness. There have been many theories about anesthetic mechanisms, and that's just a glimpse of the many proposals from ascending arousal system and thalamic switch, unbinding of brain areas from each other, gamma synchrony, and even quantum effects. Now, I'm going to focus on something slightly different, and that's dynamics. And in this slide, I'd like to first show some of the main ideas, just to summarize them, what I'm going to talk about. So the first point is dynamics. So first of all, the stream of conscious experience consists of an uninterrupted sequence of dynamically changing brain states. Now, the repertoire of brain states is my other focus point. And what I mean by that is the richness of experience, conscious experience, depends on the diversity of distinct states that the brain can assess over time. I will also touch upon the idea of criticality, or sometimes called self-organized criticality. The brain appears to be operating at the edge of chaos, and this property actually is thought to maximize the brain state repertoire. And finally, as I mentioned, anesthesia is a key focus of our interest, as we hypothesize that anesthetics erase consciousness by disrupting the state dynamics of the brain, reducing the repertoire in information processing in neuronal populations. So it's going to be somewhat technical talk, but I'd like to start first with a movie. So what you're seeing here is neuronal activity recorded in the visual cortex with chronically implanted electrodes. There are 64 sites in this picture, And as the light goes from blue to red, that indicates increasing neuronal activity. Now this is with an electrode that's implanted in the red brain in the visual cortex, and it's spontaneous ongoing activity with no stimulation. It's just a resting activity of the brain. And as you can see, it's fairly diverse. There are some hot spots. I guess that's the pointer in the middle, right? Is it the middle? Yes. Thank you. There are some hot spots that are very active, and then some other areas that periodically become active. But overall, there is a very intense neural activity without any kind of stimulation. I'd like to show you what happens in anesthesia. So we'll put this on the side, and this is the same electrode recording in anesthesia. Now, actually going to run both of them simultaneously for you, but it's easier to perceive the difference. So here is the fully awake condition, and on the right is the same electrode array in the anesthetized condition at the point where just beyond loss of consciousness. And perhaps if you see it for a couple of seconds, you perceive already the major difference. Well, the similarity, of course, is that there is a fairly high activity in the anesthetized unconscious brain also. But the activity becomes intermittent. Occasionally, there are some gaps that you can perceive when activity stops for a second and returns, as opposed to the awake condition where it's fairly continuous. So what I'm trying to illustrate is that there is a difference in the temporal domain, not so much the intensity of the neural activity, but the intermittency that sets in under anesthesia. So we return to the slides. So this is in a more accurate form. You can also summate the activity across the whole electrode array and calculate the power of neural activity. And what we depicted here is the periods when activity is high and activity is low or silent. So these little boxes are the on times. And this is in the awake animal. And these are animals anesthetized with isoflurane, which is a very common anesthetic, at 4 different doses. Now, 0.8% isoflurane is well known to be the concentration that just removes consciousness. So you can see that from the awake and the slightly or moderately anesthetized to the fully unconscious condition, what appears is the longer and longer gaps appear in the activity. And these gaps exceed 200 milliseconds, and sometimes longer, can be as long as a second. But 200 milliseconds, I emphasize, is critical because we think that is the duration of a conscious perceptual frame. So what happens is more and more frames, conscious perceptual frames, are falling out from the stream of consciousness. At least that's the hypothesis. Now I'd like to switch to the other focus, which is the repertoire. And this really comes from the theory of our colleague, Giulio Tononi, who suggested, whose theory is the information integration theory of consciousness. He has talked at these conferences, obviously, in the past. And so the main idea is consciousness is integrated information. Now, this requires 2 things. One is that there is a large repertoire of diverse, distinct brain states, which determines the amount of information. And then it also requires that these states are causally connected. And this is what brings in the integration. So that's why information integration. Now, subsequently, we suggested with my colleagues, Alkire and Tononi, that anesthesia may actually work or remove consciousness by either reducing the repertoire of states, so reducing the amount of information, or by disrupting the causal relate and the causal connectivity of these states that is directly affecting integration. So therefore, we're looking at, we hope to quantify the repertoire of brain states under awake condition and anesthetized condition. Now, this is a difficult topic, and I'd like to address, first of all, the problem. I don't know if we can shift this a little bit, but you understand, spatial scale. I mean, the problem is at what scale do we want to measure? What scale do we have to measure brain states? Obviously, brain states could be looked at molecular level to synaptic level of single neurons, maybe population of neurons, each neuron being a bit, either on or off, firing or non-firing, or it could be local populations, could be brain circuits, could even brain regions. What qualifies as a brain state is an open question. And the same issue is on a temporal scale. We're talking about nanoseconds, microtubular states, talking about microseconds, or maybe hundreds of milliseconds. 100 milliseconds, by the way, is what is often called a microstate, when EEG takes up specific configurations in the brain, or even seconds if you think of fMRI. In any case, if you would assume that we quantify brain states at the neuronal level, then considering there are 10 to the 10 neurons in the neocortex and each one has 10 to the 4 synapses on average, and each neuron can be either on or off, fire or not fire, that is binary, that would be a binary characterization, then it turns out that there would be 10 to the 37 neocortical states possible. That's an enormous number. We obviously cannot measure this. So we need to simplify the problem and define a much coarser scale of inquiry, both spatially and temporally, where we can make at least some estimates and compare the state repertoire in awake and anesthetized. So I'm going to talk about some fMRI data first and then some neuronal data. Hopefully I'm still in time. So what you're looking at is a correlation map of the human cerebral cortex. This is taken from a functional magnetic resonance image of 20 human volunteers. BOLD stands for blood oxygen level dependent. It's a basically well-accepted method of getting brain activity or characterize or approximate neuronal activity from hemodynamic signals. Now, this is taken in a task-free resting state, which is a common way of imaging the brain, and you will, I'm sure, hear more about this from Professor Lotte's talk later today. So basically shows you this color image of the brain from the lateral side on the left side, and the brain surface only is shown, is that, and you don't see the color bar because it's left off, but the red means very highly correlated regions, and yellow is less correlated, blue is even less correlated. So these brain regions are correlated highly with the rest of the brain. Now we take these data as the starting point for a computer model of the brain states that the brain can acquire over time. So that's just the input data. And just to show you, as I said, this is an average of 20 patients, subjects, how different each individual is. And you look at this as the actual correlation images from 20 different individuals, and you can appreciate how different a resting activity is. Now, that might be because these patients or subjects are thinking about something else while they're being scanned, or it could be that they have their own personal signature of brain connectivity. And it's been debated for a while how much variability is due to inter-individual difference and how much is due to just ongoing fluctuation in thought or task. There is no task in this case, but there is spontaneous thinking, obviously, which you cannot exclude. But in any case, you can use the data from all 20 individuals and look at the differences. Now, let me talk about now the model. So these are input data to a model. Some things I have to skip here. The model we're using is a spin glass model of state dynamics. And spin glass model is probably very familiar to physicists. It's a generalization of the well-known Ising model of ferromagnetism. And it's a very nice model that allows to study the dynamic interaction of local regions. So imagine here on the right that these little patches, the red patch is the same as I showed you in the movie before. These may be active brain regions, and the blue is inactive brain regions. And the assumption in the spin glass model is that global states, in our case brain states, evolved only due to the interaction of these small regions. You can call them mesoscopic regions because of the size, about 20 millimeter in diameter. The interaction with their neighbors. Now, spin glass assumes only interaction with immediate neighbors, but we assume long-range interaction. And I'll show you that in a second. So basically, a model does that every local region can be either an upstate or active, or a downstate or inactive state, and these activity states will flip with a probability that's determined by the interaction with all other regions. And then a very important parameter is the activation level, which we can control in the Ising model. That's temperature, of course. In a brain model, that's the ascending activation or arousal level of the cortex, which we can regularly adjust in a model. And obviously, a high arousal would mimic the awake state, and very low arousal would be anesthetized state, or extremely low arousal would be completely silent brain. You can appreciate here the interactions. Now, what this shows is the neighborhood or the partnership, rather, of each local mesoscopic region. So let's take, for example, these blue colors. Every color is a different cluster. So these blue patches here indicate those regions that are in correlation or interaction with each other. Yellow is another group. This teal color is another group that only interact with themselves. So they're relatively long. There are some very long-range interactions. Now, all this comes out of the input data that I showed you. Now, here's the simulation. So this is what you see real time when we run the computer in the awake state. Awake, of course, is simulated by high activation level. Let's hope it runs. So it's a little intense to the eyes, and it's somewhat sped up. So basically you see every little region fluctuating between up and down states. But more importantly, you see some large clusters forming. For example, here is on the lower right now, and that's high brain activity. It's going to shift to the prefrontal cortex, then it's going to shift back. Now it involves the Rolandic area. Now it's gone back to the temporal lobe, occipital lobe, and it keeps going. So the system forms metastable states in which it's relatively stable for a short period of time and switches to another. And each of these metastable states can be counted. I mean, all of them can be counted and then analyzed as an index of the state repertoire. I think the next one shows the same thing at low activation levels, which we think mimics anesthesia. Again, you have local fluctuations, but now these large clusters are frozen in. So they don't move. That is as if the movie or the stream of consciousness has been stopped. There's occasionally a shift to a different configuration. So now this brain in temporal, occipital, or parietal region is active, but then it doesn't move. So brain is frozen in. It's locally active, but the large-scale configurations are frozen in. If you look at the variations of one gas on the different large-scale configurations, there are quite many. And this just shows the top 40, the 40 most frequent brain states that the simulation predicts. And they're very distinct from each other, but some actually, there's some similarity here. But you see there is a subregion here in frontal lobe and also in a parietal lobe that's different. And there are, in fact, an enormous number of these variations, and these are the ones that are most frequently there, and they are metastable. Actually, that's a diagram, shows about more than 200 out of 10,000 simulation runs. More than 250 is the maximum number, and then it exponentially falls off. And then perhaps this is the most important message from the simulation. And what that shows is how the activation level affects the clustering of these activity regions. And this is the high activation level that would correspond to the awake brain. And what I plotted here is the correlation coefficient of each state with each other. So then you get a matrix. There are 10,000 states stimulated, and so 10,000 by 10,000 states gives you a matrix of correlation, a correlation matrix. Diagonal, of course, is self-correlation. So you see in this highest activation level that the correlations are relatively low, except on the diagonal. Now, at an intermediate level, there are clusters for. These are along the diagonal. These are states that persist. So the off-diagonal high levels, shown in red, indicate states that persist. And then if we go to very low activation level, which is deep sleep, then you get hypersynchronous regions. Now, hypersynchrony is something that's seen in deep sleep, in coma. It's, again, a state in which very little is occurring. So when states froze in, as I showed. And just in the interest of time, focus only on the right side of this diagram. So here we have the final result. And what is plotted here is an index of the state repertoire that we call temporal dispersion. It's a very simple formula. It's 1 minus the correlation coefficient of each state with each other divided by the variance of the correlation coefficient. So what you see is a typical second-order phase transition diagram in which as a function of X, we see the dispersion, a measure of state repertoire, as a function of the activation level, has an extremal value, indicating that brain repertoire is maximum at a specific intermediate level of activation. And we think that is actually the active, normal, vigilant brain state. If you reduce the activation level, overall activation level, then that reduces the number of states. Also, if you hyperactivate the brain, that will also reduce the state, the diversity of states. So I'm just summarizing what I've shown. So basically the dynamic repertoire of the brain is diminished when the activation level is reduced. The repertoire of these metastable states is maximal at intermediate activation level, and that the brain operates apparently at this edge of chaos, at this optimum point, which may indicate that it has the property of self-organized criticality.
20 more minutes.
20 more.
Minutes? No? Okay, that's great. So that was computer simulation, but we would like to confirm this with real data. So it's hard to, the enumeration of this repertoire is difficult because you need to scan individuals for a very long time, at least for an hour. So instead of doing that to human volunteers, which is not really like to be motionless for a whole hour, we do this in rats. So we have these long 1-hour recordings in which we scan at very high speed, scan their brains, and then determine the configurations of their bold activity. So here you see some snapshots of brain maps from rat, and there are 80 global states analyzed here. And these little numbers just show actually the ID number, the first brain's global state, the 13th, the 35th. Anyway, it's just some examples of what kind of configurations one gets over time. And this is obtained with one method that's called the regional homogeneity method. There are many methods for brain mapping. Again, this is one in which the method is based on correlation of each voxel with all of its neighbors. So when the color is bright, that is red, that means a highly correlated region, and when it's low, it's a low correlated. There are other methods, independent components analysis and so on. And we use the second method that gives us an even higher temporal resolution, approximately 1 second. And these are examples of that. Instead of that, I'm going to show you a movie. So what this does is it shows the crossing of the bold signal. When bold signal crosses a given threshold, threshold is taken as 1 standard deviation in this case. Okay, it works. So this is not a human brain, this is a rat brain, and this is the front of the brain, this is the back of the brain, so this would be occipital lobe. This is the olfactory bulb, and this would be frontal cortex, and that's the top dorsal surface, and that thalamus would be somewhere here. It doesn't really matter at this moment. So what you see is a very dynamic activity. So again, it shows red color or brighter color shows when the blood oxygen level dependent signal exceeds a given overall threshold. And what you see is a general fluctuation, but super added to that, there are these large-scale coherent activations that are reminiscent of, I believe, that Professor Mala called ignitions in his preparation. So periodically, and this is, again, this movie is slowed down a little. That's over. But you've probably seen those, right? I just run a little more. So those large activations. Now I'm going to show you this only occurs in the awake brain. All the global brain states, how do we characterize the repertoire? One way is to look at the temporal variance. The larger the temporal variance of states, the more states there are. So here is, with the first method, the regional homogeneity method, the temporal variance of regional homogeneity plotted against each voxel in the brain. So it's a probability distribution, and the yellow, the curve over the yellow region is a low-dose anesthetic in which the animal is still conscious, and in this case we use propofol, Michael Jackson's favorite. And then we go to a high dose at which the subject loses consciousness, and that's the red region. So in all cases, and these are 6 experiments, and in all cases you see a very clear reduction in the size of the state repertoire. Now from the second method I mentioned, I showed you in the movie the large fluctuations, the global ignitions. So here you see that in the awake brain, and they are almost completely gone in the unconscious state. And then furthermore, if we look at how the states vary in time, and you don't see the left axis here, but it runs from 0 to 90, where distinguish between 90 different global states. This curve shows, or the dots rather, how the brain jumps from one state to the other with time. And sometimes it appears that the brain would be in a metastable state for a couple of seconds, and these are 1 second increments. So for 4 seconds it sits in this, and then 3 seconds in this state, and so on, and sometimes it only dwells for 1 second. Now we put all the frequency of these states and plot it on a double logarithmic plot against the state index, get a very, very accurate power law. And again, I mentioned criticality. These power laws with a slope of approximately 1.5 suggest that scale-free organization. And here on the left is the awake, and here on the left is the anesthetized. And interestingly, this type of organization is very well preserved under anesthesia. However, the repertoire is reduced. And again, this is just a summary of the data I was explaining until now. Is with the first method, the regional homogeneity method. There's a very significant, a large reduction in repertoire. Blue is the awake, purplish red is the unconscious. Again, here with the other method, the blue curve represents the conscious, and the purple or red, the unconscious. There's a very clear reduction in the repertoire of brain states. And that's one other interesting observation from this, is that there is a very high prevalence of the repertoire in the midline of the brain. And this just shows you how the images were acquired. We acquired sagittal images. So each image plane is placed, this is the rat brain, of course, not a human brain, but there's one going through the center, and some more laterally, and these different anterior-posterior levels. So the one, and then we plot the variance, the reduction. See here, this is actually the temporal variance on the left against the image slice. So right in the middle, you have the highest temporal variance of states. And then with anesthesia, this is the reduction of post-anesthesia versus before anesthesia, the largest reduction occurs in the midline. And why this is interesting, because of the concept of the default brain network, which of course is a concept originating in a human brain, default network by Reichel and others, who showed that that is a network most active in a resting state, while there is no stimulus-related activity. This also is true to the rat and other animals, that there is a default network, and we get this the largest reduction in a state repertoire just in that area of the default system. And then just a few more slides. I'd like to return to the neuronal level. Because at neuronal level, obviously, we have higher temporal resolution, and we can look at state at a much greater frequency than with fMRI. So one of the phenomena is neuronal avalanches that is of great interest. So with the same electrodes that the very first movie I showed was neural activity, unit activity measured in red brain cortex, and it was with an electrode array like this. 8 contacts on each electrode, so 64 recording sites total, and that's inserted fully in the visual cortex of the rat. So from each contact you can record units, but you can also record local field potentials, which is like micro EEG. And that field potential can be used to quantify brain states as well. And if you record from all these, you see just one example here, a fluctuating signal. And Plants and group who introduced a neuronal avalanche concept propose that we can quantify these fluctuations by looking at the negative deflections in the local field potential, which actually correlates with the neuronal firing. And so these points, they indicate those when the signal goes below 1 standard deviation. One can use 2 standard deviation or anything, 3, even 4, and get the same phenomenon, but we just used 1 SD here. So these are the points where you get a very negative going LFP, which actually indicates increased neuronal firing, unit firing. And that's my last movie, just to show you what it looks like. So in this array, exactly mapped here, so that's the top of the brain, and this is the bottom, the dural or subdural electrode sites, and these are at the bottom of the cortex, and 8 shanks, electrode shanks. And I guess the interesting part of this is to see it's very different from unit activity. These are LFP obviously reflects population of neuron activity of populations, that again clusters are formed. So it's not random, but from time to time there's just a cluster down here, then up there, and it just repeats, goes around, but it's always a cluster, a group of populations that on, and these are the neuronal avalanches. So now we quantify these, and I plotted here those events when the LFP goes below 1 SD threshold, and these are the 64 recording sites, and you can see these vertical groupings of negative LFP events that sometimes last in time. Sometimes they're brief, but sometimes they're longer. It's like that, 1, 2, 3, 4, 5, 6, 7, 8. These avalanches form neuronal population activity, which also follow a power law, suggesting criticality or self-organized, scale-free behavior, scale-free organization. But most importantly, and a little bit left off, one can quantify the information that these population events carry. And entropy is one. Another measure is the known as complexity formula. So basically, you treat these coincident negative LFP, local field potential events, as a information code, like a population code. So every configuration, when this is this one, this event, this event, this event, this event, and so on, there are zillions of possibilities. You can quantify. Each one is treated as a characteristic population event. You calculate the entropy and subtract this shuffle-corrected data set from that. What we get, it's my last result, is there is a very significant and very large reduction in the information measured by entropy with the increase in anesthetic. And here we use a different anesthetic. Desflurane is similar to isoflurane. It's just another inhalation of volatile anesthetic. Unconsciousness is reached at this point at 6, well, actually slightly before, about 5%. So by the time we get to unconscious level, the information content is reduced by approximately 70%. So that pretty much concludes what I like to say. So basically what we learned is anesthesia disrupts the dynamics of neuronal activity, both with unit activity and local field potential. The brain organization appears to be scale-free, and which is indicated by the power law. And this is, interestingly, fully preserved in anesthesia, and both fMRI and field potential studies confirm that. So this is something not related to function in a sense. In a way, anesthesia works. And I think this is actually interesting to find something that's resistant to anesthesia and then find something that's not. Because it would be just way too simple if anesthesia would suppress everything in the brain. It's not the case. There's very well-preserved activity, but there's some selective effect. So the scale-free property is preserved. However, the repertoire of brain states and the information capacity are both very significantly reduced. And again, both fMRI and LFP show that. One more minute.
Yes, one more minute. Yes, we have 2 minutes.
2 minutes. So that takes me to my very last point. And that's my kind of go on the fringes a little bit. But what else can these data suggest? So this is what I called in a previous paper the forgotten present. It just goes after Adam and remembered present. So in anesthesia, could these data mean? So obviously what thing we see is under anesthesia, neuronal activity becomes intermittent. And these gaps go beyond the 200 millisecond assumed perceptual frame. They actually some gaps are very long. So James's stream of consciousness assumes an uninterrupted sequence of perceptual frames, just very much like when we see a movie played back at normal speed, we see smooth motion. The movie is slowed down, we see jagged motion. We no longer see motion, but we see disrupted images. So how do we get the continuity and the uninterrupted nature of our perception? And here, by the way, James talks about the specious present in this regard, that every frame is really, the present represents both the past and he also said the future, but basically the frames are bound, temporarily bound, in order to present, create experience. So now what we already know, anesthesia affects the temporal dimension. Anesthesia slows down the brain's clocks. It slows down the patient's response. It slows down, slows down EEG quite a bit, affects high gamma activity. It slows down thalamocortical transmission. It lowers the flicker fusion frequency, and so on. So there's an established temporal effect. What we add to this is these gaps in activity, which could be interpreted as drops. So whenever there's a skip, it is equivalent to drop frames in a movie. So on one hand, the movie is slowed down, and also some frames drop out. So the experience becomes discontinuous. And what we think that this may prevent the temporal integration that is a necessary condition. In other words, it's almost like you perceive and then but you forget it immediately in the absence of binding. You're in a constant ongoing state of amnesia. So that's where the word is. Maybe unconsciousness is none other than a forgotten present. So many people helped me with these studies, especially Dr. Liu with the red studies, and Philae and Jeanette and other doctors, and his group of people, Dr. Humphreys, Binder with the human imaging, and so on, and some support, of course. And I thank you very much for your attention.
Questions?
I wonder why you call complexity the things you define, because complexity is known. The thing defined by Kolmogorov and Chaitin in 60s in theory of algorithmic information. While what you quote, I see it's kind of normalized or relative entropy, yes? So therefore, to be precise, maybe not to use the word complexity, you or the authors you quote.
Yes, of course, I agree with you. So there are a number of measures of complexity. And this is just one that our colleague we collaborate with. He actually calls this interaction complexity. And the measure, this interaction complexity, as well as the entropy measure I showed, I didn't have enough time to explain. So it's entropy of the data stream minus the entropy of the same stream shuffled. So both measures end up measuring the portion of entropy that's associated with the interaction of the units in the system. So it's not none other. Both end up measuring pretty much the same quantity, entropy associated with interactions. That we had an interest. I mean, part of our hypothesis is that interactions matter. That's why we use this quantity. But obviously, there are many others we could try.
Next question from there.
Tony, that was excellent. Thank you very much. Two questions. It seemed you were saying that the default modes are more sensitive to anesthesia than the others. Is that correct?
That is correct, at least with this measure.
So, and we know from Rakel's work that the brain switches between default mode and sensory-based. And we know that under anesthesia, the sensory-based remains intact because we do evoke potentials.
Yeah.
So could your gaps in the gamma or in the brain activity reflect the fact that what's being taken out by anesthesia is the default mode, which is correlating with consciousness, preserving the sensory-based, which in this case at least is not conscious?
Yes, I fully agree with you. It's very possible. And obviously, to better answer your question, what we also need to analyze data with sensory stimulation and see if that is preserved. We already know from fMRI that brain remains reactive to all those sensory stimuli. Just haven't quantified the effect on the repertoire or the gaps. But yes, I believe the main point is anesthesia takes out the default.
Okay. The other point is this scale invariance is very interesting, and you showed it's preserved. The hierarchy that gives us this scale-free invariance isn't really understood. I think of it in terms of, for example, size and temporal scale going from global brain states down to the level of small networks and neurons, and then intraneuronal into the cytoskeleton, for example. So do you know, or is there any way to know whether the scale invariance under normal circumstances goes into the cytoskeleton? And if that's the case, could it be that that's taken out by anesthesia and everything at the neuronal level up continues?
Well, unfortunately, I don't have data for that, but it seems, though, that the lower you go on a scale, the greater the effect. So by extrapolation, I would think probably your cytoskeletal system is more affected than the large-scale systems.
Thank you.
Very nice data. I want to make one comment and then ask a question. One comment is about what goes out in anesthesia. It could be the default, but another alternative, it could be high-order areas of processing. So you could get sensory responses from early processing, but it's still, there is some evidence that high-order levels in the sensory system are going out. And the question is about the repertoire. It was not clear whether you saw a reduction in the repertoire under anesthesia when you did your avalanche analysis, and whether you did it also in the gamma power modulations, this avalanche type analysis in the rat. And the last slide that you showed, was there also a change in repertoire, or how is the information measure related to that?
Yes, thank you for the suggestion. By the way, we also find that the higher order regions are affected in other models. But obviously this work is ongoing, and the data hasn't been really sorted out in terms of regional effects, because you could also go anterior-posterior divisions, and we haven't done that. Yes, the It was actually gamma. That avalanche was from LFP filter to 20 to 80 hertz. So both the information and the avalanche are gamma, which is not a traditional way of looking at avalanches by planes and others. They filter down to 3 hertz and up. And that's a difference. You only see this effect in a gamma ray.